Let's start by comparing prokaryotes, eukaryotes, and viruses. Eukaryotes have a nucleus and organelles. They can be unicellular or multicellular, and they usually have several chromosomes made from DNA. Prokaryotes have no nucleus, little internal organization. They are always unicellular, and most have one circular chromosome, which is, however, always made from DNA, just like in eukaryotes. Viruses, on the other hand, have no cellular organization. Their genome is very small and can be RNA or DNA. Both can be double or single-stranded, and they are not considered alive because they don't have that cellular organization. Viruses are considered obligate intracellular parasites. They cannot reproduce on their own. Obligate means exactly that, and that is one other reason why they are not considered alive. Intracellular because they can only replicate inside of host cells, and parasites because they derive a benefit at the host's expense. Someone has called, and I don't remember who, a virus a piece of bad news wrapped in protein. I found that a very good description. So there's a genome that is surrounded by a capsid, and the capsid is composed of proteins. You can see these different capsomere subunits forming a capsid in different viruses here in the figure on the left, and then below you see the micrographs. Some viruses also have host-derived membranes. So when they enter the cell, they just take a piece of the host membrane with them, and really especially mean first they make the host express some membrane proteins on that piece of membrane. What I mean with acellular is that there's no energy metabolism, and again that they are obligate intracellular parasites. Viruses have a host range, and what that means is the type of organisms or cells that a pathogen can infect, in this case the virus. And this is usually determined by the receptors on the host membrane that allow the virus to gain entry, or at least allow the genome of the virus to gain entry into the cell. So the cell surface molecules determine the host range, and the host range is usually rather narrow. The viral life cycle usually has 4 stages. It starts with the entry and uncoating of the DNA capsid. Sometimes viruses also can enter through endocytosis, and as of today, it wasn't known how the coronavirus, for example. The second stage is the replication of the viral genome. The transcription and manufacture of capsid proteins is third. And then viruses self-assemble new virus particles and exit from the cell. In the case of envelope viruses, as I said, they take a little bit of the viral host envelope with them. Again, entry and uncoating, replication, transcription of capsid proteins, self-assembly, and exit. The influenza virus, which we will discuss today, has a small genome composed of 8 individual RNA molecules, and they have 2 antigenic glycoproteins on the surface. What that means is that these glycoproteins that stick out from the virus can cause an immune response of the host. There is a hemagglutinin molecule The gene has 16 different alleles, and that is responsible for attachment to the host. And there are neuraminidase molecules that have 9 different alleles, and these are responsible for passage through the mucin, and the budding. That is where the names H1N1 derive from. That it's the one allele of hemagglutinin and the one allele of the neuraminidase would be an H1N1, and H9N1 would be the 9th hemagglutinin allele combined with the first neuraminidase allele. The SARS-CoV-2 virus, the novel coronavirus, is very similar. It has spikes called S spikes that bind to the host receptor. They are so dense that it gives a halo, and that's why it's called a corona, like a halo around the virus. And structural biology and bioinformatics is really, really important to understand this binding to the host cell so that you might find molecules that block that binding. There are different types of influenza viruses, just like there are different types of coronaviruses. There's A, B, C, D, and they're different from each other in which host they can infect. The one that we are mostly concerned with in the influenza is type A, because it has a broad host range: humans, birds, swines, and horses. In SARS-CoV-2, it's alpha, beta, delta, and gamma. Only the alpha and beta coronaviruses are known to infect humans. 7 coronaviruses have been identified. The deadly ones are SARS-CoV, which causes the severe acute respiratory syndrome in 2002, the MERS-CoV, the Middle Eastern Respiratory Syndrome, that appeared in 2012 and is still ongoing. So I find a very interesting comparison chart of the different respiratory endemics. Take a little while if you are interested in it to look at this. So on the left side you have the fatality rate on the y-axis, and is a log scale, and on the x-axis you have the speed of spreading, and you can see where the Spanish flu, the SARS, the MERS, the common cold, the chicken pox, and the new coronavirus fall.